Dear colleagues, let me present it for you the report from the collective of authors called Portable Nose for Diagnostic of Inflammation and Diverse Variation in Health Status of Humans and Animals. The presenter is Anastasia Shuba, the Assistant Professor of Physical and Analytical Chemistry Department in Voronezh State University of Engineering Technologies. The more detailed biography is shown on the slide. Our research group are working on the development of the new way and devices for diagnostic using gas phase analysis by sensor array, including search and development new selective paths for sensor covering, also mathematical treatment of multivariate output data of sensors, including improving precision and accuracy of measurement, also the development of program for sensor devices. The fundamental problem which we try to solve in our investigation is connected with metabolomics. On the slide presented the metabolomic map from famous site with metabolomic pathways, including conversing of macronutrients with micronutrients in simple organic substances. But producing of highly volatile compounds is not clear, understandable, and not presented on this map. We try to fill up this gap and using this knowledge for clinical diagnostic. Why we're interested in volatile metabolome? The first is that volatile metabolome has the most fast and instant changes in the organism, including after influence. Also, some biosamples investigated with gas chromatography and mass spectrometry, and volatile metabolome partly is known. Also, the most pleasant for diagnostic is that volatile metabolome can be measured easy and non-traumatic. Based on these advantages of volatile metabolome, the technology of nose is spreading in many directions of medicine, including pediatrics, narcology, endocrinology, immunology, gastroenterology, and so on, investigation of infection disease, and the most wide direction using nose technology is clinical diagnostic. If we look at the old techniques and approaches to the clinical diagnostic of organism pathology, that it could be divided on three groups: biochemical, chemical, or physical tools. Each group has pros or cons for clinical implementation. In connection with previous information, the goal of this research is development and application of a new mobile device based on piezoelectric sensors for assessing the health status of organs and systems of humans and animals by analyzing the volatile metabolome. Our group on these days developed the two variants of electronic nose commercially available. The first variant has developed for analysis of equilibrium gas phase over different samples. The second one for frontal input gas phase into the detection cell. Both devices are working on the piezoelectric sensors and connecting with a laptop or PC for recording and processing data from sensors in special software. The both variants of nose operating in the ambient temperature from 15 to 35 degrees of Celsius. It should be noticed that permissible temperature variation during one research is not more than 3 degrees of Celsius. The optimal conditions for investigation of BUSA are stable temperature and humidity, and the weight of devices is not more than 400 grams. The selective phases of piezo sensors are chosen among 50 sorbents based on previous experiments of sorptions of different classes of volatile substances, biosamples, and food products, and so on. The piezo sensors with these coverings are stable in working during long period of time, up 2 years for nanostructured solid phases in open detection cell, such as multivolt nanotubes, nitrate of zirconium oxide, biohydroxyapatite. Piezo sensor with these coverings stable at the intensive work. Exploitation minimum 10 hours per day. The fast sorption and desorption processes on these sorbents allow to minimize the time of measurement. The primary output data from sensor array is chronofrequency grams. This is dependence of a difference of piezo sensor oscillation frequency comparing with the value of base oscillation frequency corresponding to frequency before interacting with vapors of substances. The simplest visual information is presented in form of visual prints in two variants: visual prints of maximum and kinetical with visual prints with different algorithm of building. The square of visual prints of sensor signals in both variants is quantitative characteristic of volatile compounds mixture. Our group proposed the parameters for qualitative analysis of gas mixtures. This is parameter of sorption efficiency, AIG, calculated using the maximum signals of sensors during sorptions of volatile organic compounds in gas phase over biosamples or substances. The sensor array with the proposed sorption phases are trained by 55 individual substances of normal and pathogenic metabolites, including alcohols, ketones, aldehydes, carboxylic acids, amines. Early, using the statistics of binary responses, we established the values of parameter of sorption efficiency for identification of substances. It should be noticed that values of these parameters depend on way of input of gas phase into detection cell. We estimated these parameters for injections of equilibrium gas phase and for frontal spontaneous evaporation of gases from biosamples inside detection cell. We demonstrate the application of portable diagnosis into clinical diagnostics of disease in animals and humans. And the first application is diagnosis of respiratory disease in cows. In this research, the main task was find out the calves with bronchopneumonia which need medical treatment and, if possible, to separate calves with subclinical course of respiratory disease to prevent the development of pneumonia by signals of sensors array. We investigate 65 calves in age of 3 to 60 days of life, and the biosamples for analysis by portable electronic nose with frontal input of gas phase were condensate of exhaled breath and nasal mucus. To establish the health status of respiratory system of calves, we used the common clinical methods and VI score system, also special methods such as spirometry with functional probes with loading on respiratory system, and laboratory methods. The results of clinical and laboratory analysis of calves' respiratory health status were used to estimate correlation with parameter of sensors array and to establish the correct clinical diagnosis. Based on the results of correlation analysis and algorithm of choosing important variables in multivariate regression, we constructed the mathematical models using projection on the latent structures for the prediction of the most informative clinical and laboratory indicators for the correct diagnosis. This is prediction of VI score and bacterial contamination of bronchoalveolar lavage. Consistently applying these models to result of analysis of case phase over condensate X-hold breath samples by portable electronic nose, we can identify all three clinical groups. Using the parameters AIG, characterized by statistically significant correlation coefficients with clinical and laboratory indicators, and valuable for regression models, we identify the volatile markers of respiratory disease in calves. The number and composition of case phase over biosamples is different and depends on metrics of biosamples, presence of microorganisms, products of inflammation, and immune response. For example, for nasal mucus of calves with symptoms of damage of respiratory organs is characterized by heavy cyclic substances as containing compounds due to presence of pathogenic microorganisms in nasal mucus. This assumption is confirmed by monitoring of samples of nasal mucus during a day. The results show influence of health status of calves' respiratory tract on the case phase over nasal mucus and could be used for prediction of disease for one sample without complex mathematical processing. The second approach of using portable nose is measurement and identification of volatile metabolome of individual person and search the features for monitoring and preventing development of disease. Nowadays, our database contains 9,000 measurements and is updated every day. We proposed measuring the volatile metabolome by the forearm area of the skin using portable nose with open detection cell contacted with this area. Analytical signals of the array are four multidimensional visual prints: general state, energy, endocrine, negative. Each of these visual prints represent a certain more differentiated part of the chronofrequencygrams of all of the most informative sensors. They differ in the set of fixed response times of each sensor and the number of sensors. The largest number of points on the visual print correspond to the general state, all 8 sensors, and 7 time points on the chronofrequencygram. Mask and a set of sensors for constructing multidimensional visual prints were selected based on the results of preliminary training on individual substances that correspond to the malfunction of metabolism in individual organ or inflammation. For example, ketones, aldehydes, acids, hormones characterize the malfunction of the pancreas and gallbladder. For them, an endocrine mask was built while the signals of 3 sensors were selected. For energy mask, signals of 3 sensors were chosen at desorption part of the chronofrequencygrams, which is the most informative for part of metabolome connecting with synthesis of ATP and physical actions. For negative mask, signals of 8 senses at the first time of sorption and part of desorption were chosen, which reflects the severity of destructive processes in the body by present cyclic compounds, heavy ketones, and amines. Also, we established that comparison of visual prints for left and right hands is significant for diagnosis of descriptive states such as stress, pain, spasm, and so on, and for some pathological states and disease. For example, for mask of visual prints energy after meal, unlike the normal, the value of square of visual prints for right hand is more than value of square of visual prints for left hand, up to 30%. The difference depends on intensity of digestion and enzyme production. We noticed that the changes of parameters values RIG are also informative for different health status, such as stress, weakness, exhaustion, and clinically approved conditions such as inflammation, hormone imbalance, high level of glucose, infection, virus. Each parameter has a color regions depending on parameters value. Moreover, the critical and clinically significant changes in metabolome reflect in red and burgundy colors of parameters values. For visualization of this information as understandable for any users, we developed the additional program for construction of sphere of health status. This sphere is constructed by parameters A, I, G values. The more green zone on this sphere, the more health status close to the normal. For yellow, red, and burgundy zones, the window of program shows description of possible causes that can affect health. Let's consider the diagnosis of the metabolism disturbance in children. We investigated 20 patients during treatment in the hospital. The table shows diagnosis some of them. The propandial and ethandial were included in the set of substances for the training of the sensors array. Because the ethandial is the metabolite of glucose in the body, and we feel hunger when level of glucose decreases in the blood. The propandial is lipolysis metabolite, and its high level in blood can be at prediabetes mellitus condition. This is the result of monitoring hypoglycemia treatment. At first point of monitoring, 6 parameters had abnormal values corresponding to hunger, inflammation products. After meal and treatment, the one parameter stayed in red zone due to variation in hormones that normal at this age. Another example, it is monitoring of dynamics treatment of viral infection with bacterial pleuritis. The sphere of health status shows critical changes in volatile metabolome before clinical symptoms for red zones. During treatment, the metabolite of antibiotics also identified in volatile metabolome, but inflammation products and metabolite of bacteria is not identified at the end of the antibiotics treatment. The more deep clinical diagnostics by volatile metabolome can be provided on the Zaharin head zone. For example, monitoring zone of lungs during pleuritis treatment. The results of this monitoring correlate with results measured on the forearm. More red zones on the sphere of health status are connected with diverse inflammation products after active bacterial infection. Generally, we propose diagnostic of 17 health status, including disease of skin, prediabetes, diabetic conditions, ketoacidosis, bronchitis, pneumonia, and clinical diagnostic by the smell of other bioassays such as blood, urine, sweat. The sensibility and specificity of some proposed methods based on the comparison with diagnosis by traditional methods in clinical diagnostic are shown on the slide, and these are higher than 70%, that is appropriate for test methods. At the end of my report, I thank you for your attention and answer for all your questions below this video or by email. We're glad to cooperate with other research group. Let me wish you take care of yourself and be healthy. Goodbye.